starting has made this much polypeptide. So polyribosomes are found both in prokaryotes and eukaryotes, and they help in increasing the efficiency of the protein expression. So this is all we have discussed. This is just a summary. You can go over them. And the last topic I would like to talk in this chapter is mutations. Now, what are mutations? Mutation is a sudden change in the, or it's a random change in the DNA. Okay, so mutations are changes in the genetic information of a cell. Now, there are some, we are going to talk about different kinds of mutations. Point mutations are those mutations which are just in one nucleotide. Okay, so this is a typical example of a point mutation which takes place in sickle cell anemia. So this is the wild type beta globin protein. So this is the sequence in the wild type beta globin DNA, and this is the mutant. So in the wild type beta globin, so you know that hemoglobin has is a protein which is having 4 subunits, 2 alpha chains and 2 beta chains. So there's a mutation in the beta chain. So this is the wild type. The sequence is CTC. In the mutant, it is CAC. And as a result of that, there's a change in mRNA. In the mRNA, it is GAG, and the mRNA in sickle cell anemia is sickle cell anemia is GUG. And as a result of that change in mRNA, there's a change in amino acid. In the normal hemoglobin, the amino acid is glutamate, which is charged amino acid, and in the diseased state, it is valine. Valine is non-polar amino acid. So this is a typical example of point mutation, and it causes sickle cell anemia. Now, the mutations, they can be substitution mutations, or they can be insertions or deletions. So substitution mutations are when one nucleotide is substituted for another. Suppose it's A, it becomes G. If it is G, it becomes T. So that's substitution. Deletions and insertions means if there is a deletion or the insertion of some of the nucleotides. So insertions and deletions are more dangerous mutations because they can totally change the reading frame, and they're called frame shift mutations. Okay, some of the mutations are called silent mutations, which means they do not cause any effect. The reason is if there is a mutation and it does not affect, why it doesn't affect? Because for some of the amino acids, there are more than one codon. Suppose there are there is a codon called, suppose it's GGC, it codes for serine, and there's another codon GGG, it's also code for serine. Suppose because of a mutation, the codon has from GGC, it has become GGG, it's going to code for the same amino acid. So these kind of mutations are called silent mutations. Missense mutations are those mutations which change the amino acid. Nonsense mutations, which actually it's a kind of a mutation because of which the codon becomes stop codon. Okay, because of a mutation, the codon becomes a stop codon and it stops the process of translation. Those kind of mutations are called nonsense mutations. So let's and what the for the mutations can be caused by physical or the chemical agents, and they're called the mutagens. So chemical mutagens can be many chemicals can cause mutations, and the chemicals which cause mutations cause cancer, they're called carcinogens. Okay, now this shows you some of the examples of the mutations and the effects. Now this is the wild type gene, and this forms mRNA, and this is the sequence of amino acids in the protein. This is the primary structure of the protein. So let's see, if suppose there is some mutation takes place, substitution mutation takes place and instead of G there is A instead of G. So you can see in the wild type there is G, and if the substitution takes place, instead of G there is A, instead of G there is A, as because of that in the mRNA there is U instead of C. Because of this change there is a change in mRNA, and as a result of that, so this is GGU now. Earlier it was GGC. GGC codes for glycine. Now it has become GGU. It also codes for glycine. So this kind of mutation is called silent mutation. Now here it is T instead of C. In the wild type it is C. Okay. In the wild type it is C, but here it is T. So when there is T instead of C, in the mRNA there is A instead of G. Okay, and as a result of that, the amino acid will be serine instead of glycine. So these kind of mutations are called missense mutations. Okay, here you can see A instead of T. So instead of T, if it is A here, in the mRNA it is going to be UAG, and UAG is a stop codon, and these kind of mutations are called nonsense mutations. Here, suppose there is one extra A added. This is insertion. As a result of that, in mRNA there will be extra U, and as a result of that this codon becomes UAA, so it's a stop codon. Now suppose here A is missing, and as a result of that in mRNA U is missing, and as a result of that what happens is, if U is missing, okay, if U is missing, then the next codon is UUG. Okay, next codon is UUG. In wild type the codon is UUU, it codes for phenylalanine. Now the codon is, instead of UUU, it is UUG. It is UUG. When it is UUG, the amino acid is leucine. And after that, all the amino acids are going to be different. So because of this, A missing here, there's U missing here. After this, the whole reading frame changes. So these kind of mutations are called frame shift mutations. Suppose here this full codon, these three nucleotides are missing. TTC is missing, and as a result of that, AAG is going to be missing. So AAG codes for lysine. So here, as a result of that, one complete amino acid will be missing. So this is basically all about your mutations. So that's all about this chapter.